Hello, I'm Dongyoung Kim, currently a senior at Aju University, Department of Biological Sciences, taking an advanced bioinformatics class. For the final project, I progressed the project focused on how to better infer missing data. The presentation topic is correlated imputation between genotype-phenotype data using autoencoder and projection Procrustes analysis. In the present day, these various fields of biological sciences are the subject of bioinformatics research and can be quantitatively analyzed. From this point of view, almost all researchers in the biology field commonly deal with these metric data as input. But the problem is that the data that we actually handle isn't that perfect. It's inevitable to face these various missing values, especially in the macrobiology field due to various realistic limitations in the data collection procedures. Then how do we treat them? The statistical methodology to handle these missing values is called imputation. The most basic imputation that we can consider is to replace a missing value with a specific constant value like 0. Or you can replace it with the average or median of each variable. In the case of a correlation existed, it can be estimated by interpolation or extrapolation. In addition, various imputation methods such as KNN, HOTEC, and dim learning methods are known. They basically deal with missing values that are observed in one matrix. What I am interested in is not just one matrix, but multiple matrices. This seems to be a particularly common case in the field of biology, where you can get a genotype data matrix and phenotype data matrix for the same sample. At this time, there may be cases where genotype data from a sample exists, but phenotype data does not exist. It's actually a very common case, as I think. My question is, how do we better infer the phenotype of this object in this case? We can assume about the case like this. This is a cave-dwelling harvestman that I'm studying. One day, I took out a specimen from this specimen storage and found that there were only samples of juveniles. In this case, genetic analysis can be performed with these samples, but morphological data cannot be used meaningfully even if gathered. So I went to the cave where they collected, but it was illegally occupied and renovated by shamans in recent, so I couldn't get any more samples of it. What we can do in this situation is just to use genetic data that we have to infer the phenotypic data as best as we can. While I was thinking about this, the first idea I got was projection Procrustes analysis. Procrustes transformation is named after a bandit from Greek mythology who made his victims fit his bed either by stretching their limbs or cutting them off. In Procrustes transformation, it performs geometric transformations such as translation, rotation, or scaling without changing any data relationships or its own properties. This is the paper that I got the idea for. If you look to the left part of the right figure, there's a two-dimensional PCA result of human whole genome sequencing data. And on the right, there's a high-dimensional PCA for particular marker genes of human. We can think about that when we get a new patient. There are a lot of realistic limitations for everyone to conduct a whole genome sequencing. Instead, the paper suggests that it is possible to estimate approximately where the genetic profile of a new patient is by transforming the PCA of particular gene markers into the whole genome sequencing PCA using projection Procrustes analysis. The second idea is an autoencoder. Autoencoder is a deep learning algorithm that organizes and optimizes neural networks in a way that minimizes the loss between input and output data. Here in the figure, input and output can be a variety of data as well as pictures. In fact, there was a study in which human whole genome sequencing SNP data was dimensionally reduced through an autoencoders. Although there are many multivariate analysis methods for dimensionality reduction, this is the reason why autoencoders are special. Autoencoder can not only be used for dimensional reduction, but conversely, it can unfold the reduced values back into the matrix. Given a random coordinate on the plot, then by inserting it into the decoder, we can get a corresponding putative SNP matrix as an output. My design of combining these two ideas is this. First, I can dimensionally reduce the genetic data using PCA, and then the phenotype data can be dimensionally reduced by extracting the bottleneck layer of the trained autoencoder model. Here, let's assume that there is only genotype data of specific sample of our interest. So the number of samples would be one more in the genotype data. After that, transform the genotype PCA plot through the projection Procrustes to the phenotype autoencoder scatter plot, like this. And so the genotype data will look very similar to the phenotype autoencoder. At this time, our sample data, where phenotypic data did not exist, will also be transformed into a specific transformed coordinate. Now we are going to put the coordinates of this sample back into the decoder of the phenotype autoencoder, and then reconstruct the unknown phenotype data. For implementing this idea, I used morphological measurement of 65 variables as the phenotype and the SNP matrix of the CO1 gene as the genotype of the Korean cave-dwelling harvestman I collected. And secondly, I also used the panel data of Drosophila melanogaster that was available online for testing. The phenotypic data provided here is non-morphological, but behavioral experimental data such as aggression and chemical reactions, etc. And whole genome sequencing data is provided in this VCF form. So I downloaded it from a Linux server and converted the format with Plink and ConvertF, and then conducted a principal component analysis with Smart PCA. Then I got the result. Firstly, I will show the analysis process of cave-dwelling harvestman. First of all, the SNP site was extracted from the CO1 sequence and PCA analysis was performed. Our sample data was marked with the purple stars here. Next, I constructed the neural network for the autoencoder and then trained with morphological data. Using the keras in the R environment, I could get and evaluate the model. It's a trained model that shows 96.3% of accuracy. And within this model, I was able to get this dimensionally reduced coordinate from a bottleneck layer. After that, I converted these genotype PC scores to phenotype autoencoder bottleneck layer values. At this moment, there is one more sample of genotype PCA, so it is not the same dimension. And in this case, we cannot proceed with the general Procrustes analysis. We have to conduct a projection Procrustes analysis. This paper introduced earlier has a good explanation of the procedure, so I implemented it as R referred to the paper. Let me explain this procedure in short. Let's denote that the phenotype autoencoder bottleneck layer value matrix, that is the target of transformation, is Y, and the PCA scores matrix that is subject to transformation is X. At this point, the X matrix is multiplied by the A matrix to equalize the dimension with Y, and then multiplied by a constant value called rho, which is a constant that minimizes the difference with Y. And the rho XA matrix itself become a newly transformed matrix. By multiplying the obtained rho and A equally to our sample data vector, we can also be able to obtain the transformed coordinates of the sample data, like this. Through this, I performed projection Procrustes analysis. At this time, the quality of the transformation depends on how many dimensions of the PCA data. In other words, how many principal components were used for transformation. As the number of pieces used increased, the distribution of our target phenotype data and transformed genotype data became similar. And when I performed using 30 principal components, I got very similar transformed coordinates. So I used output that was converted from 30 dimension of genotype PCA. I could mark the transformed coordinates of our sample data of interest in the original phenotype autoencoder plot. And then I could get this estimate of the morphometric measurements by putting the coordinate in the decoder of the already trained autoencoder or phenotype, like this. The result is that this process has been implemented successfully. And the question is, how accurate is this? After I was concerned about how to verify this, I compared it with the reconstructed data through another commonly used imputation method that I mentioned earlier, that like median imputation or mean imputation. This is the ratio of the error between the real data and the estimate divided by the standard deviation of each variable. In other words, we calculated how much the error was caused by imputation compared to the difference between the data on average. I found that the data that I reconstructed using Procrustes and Autoencoder showed significantly lower levels of error than other methods. Next is an analysis using Drosophila data. The detailed process is the same as before, so I will skip it. I also got a genotype PCA and phenotype autoencoder, and it was then transformed into projection Procrustes, which also achieved relatively good performance when using higher dimensions. And I inserted the transformed coordinates of the sample of interest into the decoder of the phenotype data. And I reconstructed the phenotype values in the same way. If you look at it now, the Procrustes transformation was not going well compared to the Harvestman dataset. As I will tell in detail later, it seems to be because phenotypic data has too little relevance to genotype data. Nevertheless, the reconstructed data resulted in a much less sum of scattering error rate and error rate relative to standard deviations than other imputation methods. Now it's a discussion. I want to talk about is that idea can actually be implemented. However, I think it needs to be more advanced and verified by other statistical tests as to whether this method is actually appropriate and efficient enough to be used in the field. Secondly, I would like to discuss the difference in the performance between the Harvestman dataset and Drosophila dataset. If you compare it, you can see that the Procrustes transformation was much better in the Harvestman data. I think a decisive factor that leads to the different performance between two datasets is the distributional similarity between phenotype data and genotype data. The important thing about this analysis is not whether a particular variable, not each specific characteristic, is related to a gene or not, but whether the distribution of data in n dimensions is similar between two data. That's why the Harvestman dataset has been transformed well, even though it's just CO1, a barcode gene. It's not because it's associated with these phenotypes, but because it's a gene highly reflecting the high dimensional morphological distribution of samples. On the other hand, performance in Drosophila data was not good because the phenotype and genotype data were not related, even though genotype data was whole genome sequencing data. So I think it's necessary to check how much clusters or clades of genotype data are applied well within phenotype data first. Because of this approach, it seems necessary to conduct this analysis after checking it with your own eyes rather than automation, because there is a difference in performance depending on the data you have. Therefore, I think it is an important process to test the data that you already know the answer to and then apply the actual phenotype missed data. Basically, what I want to talk about is that it's not the best way to deal with the missing data, and it's not the absolute way to do it. The best thing is to collect the empty data directly, but in reality, there are a lot of limitations. In that case, you must think about how to infer in a more rational and reproducible way. Our goal of science may be to approach real fact, but also I think it's also very meaningful to infer the fact in the most reasonable way within the technology we have now. And finally, this is a method to get a little bit of a loophole and a lot of tackles from a biological perspective. At first glance, it can be misunderstood that phenotypes can be known by genes, but I'd like to tell you that absolutely not the case. It's definitely neither the support of genetic determinism nor the misconception that phenotypes can be explained within only genotypes. Of course, phenotypes have to do with complex factors that can never be explained solely by genes, but the goal of this project is to suggest a novel statistical framework for what's missed from the data we have as best as we can. So please reconsider this point of view again, and I will end the presentation with this. Great, thank you for listening.